Hi everyone, thank you for joining me today in this talk. Get the fast, simple, and accurate DNA quant you deserve with Lunatic. I'm sure most people still remember this sound. Of course, amazing at the time what could be done, even though it came with some disadvantages. It took a while to log in, it wasn't very fast, and you couldn't use your phone while being online. To me, that feels a bit the same as techniques and instruments for DNA quantification. Older techniques were of course super innovative at the time, but technology has become better and better. And now the original methods just can't keep up in terms of speed and simplicity. Our Lunatic is a full spectrum UV spectrometer which requires only 2 microliter of sample and no dilution steps or standard curves. Lunatic can measure up to 96 samples at a time in just 10 minutes using our plates with 2 microcuvettes, which allow reading higher concentration samples. And if you would only work with low concentrations, we also have a second plate with just 1 microcuvette that can read 96 samples in just 5 minutes. Measuring nucleic acids with UV-Vis can be relevant in a lot of steps, and mainly for one of these reasons, either to determine the RNA or DNA concentration, and ideally at the same time checking the sample purity, and before and after normalization to calculate either your dilution or to make sure at the end you have the amount you wanted. Lunatic is the only system out there that can measure nucleic acids at high concentration and high throughput. It uses a microfluidic circuit with 2 fixed path lengths to cover a wide dynamic range of 0.03 to 275 OD, and that translates into 1.5 nanograms per microliter until 13,750 nanograms per microliter double-stranded DNA. The unique microfluidic circuits molded into Lunatic plates ensure that there's no cross-contamination or evaporation. Once the sample is loaded, it's pulled into the reservoir area by capillary action. When a sample hits a hydrophobic point, it stops. At this step, the sample can sit up for up to 2 hours without any evaporation. After the plate is loaded into the instrument, the empty cuvette is read first, and that will remove any absorbance contribution from the plastic. And then a pump applies a small vacuum, and so the sample gets moved into the cuvettes to be read. Here we can see exactly what sample loading looks like with the help of some fluorescein and a black light. In 2 wells we've already added 2 microliter of fluorescein. As you can see, to load another sample you simply add in the 2 microliter into that input well and the sample will wick into the plate. Now on the left you can see those loaded samples and on the right you can see samples after being measured with Lunatic. The 2 diamond shapes are the cuvettes for one sample. What this also shows is after pumping, all the sample remains in our consumable, so there's no contact between the sample and the actual pumping system. No contamination possible. Now, how can we prove that our fixed path lengths are so exact? By using 2 path length absorbance standards for microvolume spectrometers, certified by the National Institute of Standards and Technology, or NIST. Uracil and tryptophan NIST standards are measured for absorbance and the expected value is compared to the measured value. If they're close, then our fixed path length can be used for accurate and reproducible measurements. All of the path lengths calculated based on the measured absorbance in 5 different instruments run in octuplicate of these uracil and tryptophan standards were within 1.65% of the nominal path length, and the mean, which is the x here, and the median, which is the middle line, were within 1.25%. This means that Lunatic generates data you can really trust. And here's a look at linearity, accuracy, and precision. This shows the DNA dilution series of calf-tinous DNA from 25 nanograms per microliter until 1,400 nanograms per microliter. The close agreement between the target concentration and the average measured concentration shows of Lunatic spot-on accuracy within 2% of the expected value. And the low percent CVs across the dilution range help show off its amazing precision, typically below 1%. Delivering this dynamic range, linearity, accuracy, and precision is the advantage of using 2 fixed path lengths. Now, groups producing plasmids, for example, typically work with high concentrations. I spoke with a customer recently who was getting variable results due to sample dilutions that were required with their old methods. For that group, Lunatic was a great solution because they could read samples without dilution. The data shown here demonstrates how Lunatic handles high concentration samples without dilution with the same great accuracy and reproducibility. The highest concentration of DNA in this dilution series was 12.8 micrograms per microliter. The R-squared value for the series showcases the high linearity across the huge dynamic range for Lunatic. Just run your samples at the concentration you're actually using them. Don't add in extra dilution steps you don't need. Since DNA and RNA samples can be freaking messy, I want to show you on this slide and the next one how Lunatic's unmixed applications let you see annoying impurities that other UV systems would mistake as DNA or RNA. Here we're looking at results from a simple, pure half-timeless DNA in TE buffer. This is a typical result for a clean sample. We can see the expected peak, maximum at 260 nanometers. The quality ratios are at the expected value. So A260, A280 around 1.8 for pure DNA, A260, A230 slightly above 2. But note the accuracy and reproducibility. We've seen nice low standard deviations and percent coefficient of variation for these triplicate reads. Now we're looking at the result of a DNA sample that's been contaminated by protein. In both graphs, the blue line is a total measured spectrum. Simple total absorbance data is on the left, and on the right, we're analyzing the sample with our unmixed algorithm to deconvolute the total signal into the signal from DNA, the protein, and other impurities. By using unmixed, we know exactly the problem and how to solve it. This sample would need to be repurified before using it in a downstream application. So don't miss contaminants that can screw up your sequencing. Check your preps on Lunatic and know what's in your sample. One of our customers had an issue where they were getting higher DNA concentrations by absorbance measurements compared to when they used a fluorescent dye. To troubleshoot this, they ran lambda DNA through their purification steps and compared the sample using Lunatic before and after purification. The concentrations were similar, but the sample had significantly higher turbidity after the purification, which is shown here on the right. The customer concluded the most likely source was bead carryover from the purification. Now, our unmixed app successfully deconvoluted the turbidity spectrum. In fact, the unmixed results were actually closer to their original fluorescent dye results and could be done faster and cheaper without additional reagents or dilution steps. Fluorescent protocols are multi-step, they require dilutions, they take time, use costly reagents, and can introduce pipetting errors, which negatively impact results. You will need at least 6 steps to get to your actual results, with a lot of sample prep. On Lunatic, you can just load and read, no sample prep at all, and no need to dilute to get into the measurement range. And these costly fluorescent dyes are only linear for a short range. At low concentration, Lunatic and Picogreen will agree, while at a higher concentration, these costly fluorescent dyes will saturate. Ditch the time-sucking dye-based prep, wasteful standard curves, and the risk of being outside the linear range. Know right away if your samples are good to go with Lunatic. In the experiment shown here, you can see the linearity for samples read using Lunatic across a wide concentration range. You're able to see the saturation effect with Picogreen here. This curve was generated using the same samples as for Lunatic, but the samples were diluted to 100-fold. Because of the limited dynamic range with a fluorescent method, a sample might have to be diluted multiple times so that it falls on the standard curve. You will run the sample from different parts of your dilution to make sure at least one of them falls in your measurement range. This is a waste of samples, reagents, and time. On the other hand, Lunatic offers a wide dynamic range with reproducibility and accuracy without the hassle of standard curves and dilutions. And these multi-step procedures have an impact on readout accuracy and precision. This experiment was run by one of our high-throughput customers. They measured thousands of genomic DNA samples using either Lunatic or Picogreen. Based on those results, the samples were adjusted to 25 nanograms per microliter and then read again. And you can see there's a much wider variation in the results with Picogreen compared to samples read on the Lunatic. And in the range where Lunatic and Picogreen both work, there's a good correlation between the methods. Here we're showcasing the results from the UK Biobank cohort. This was a massive genotyping project involving about 500,000 participants. Results from almost 500,000 samples show strong correlations in readouts using Lunatic compared to picogreen. And importantly, there was also no difference in the quality of downstream data. Don't lose sleep over instrument to instrument or lab to lab variation. Lunatic loves repeating itself, so standardizing your method across all labs is very easy. With boatloads of samples at multiple locations, the last thing you need is to struggle with validating the same workflow over and over again. So here you can see a box and whisker plot with 3 concentrations of DNA. So either they will run in octuplicate on one system, or they will run in octuplicate on 8 different Lunatic systems, for a total of 64 samples per concentration. We've already discussed the amazing results for one instrument, but here you can see that with 8 completely different systems, there's also nothing to worry about. Having systems in different labs in the same building, or even at different sites globally, will always provide the same high quality results. And really, Lunatic and Quantidol, high and low, always from 2 microliters. For low concentration DNA extracts where you can easily quant without any dyes or standards, to very high concentrated plasmid DNA where you just quant directly without the need for dilutions. And if speed matters, Lunatic has you covered as well. Hands-on time for 96 samples for a new user with a single channel pipette with 17 minutes to fill a plate on Lunatic. While doing this on an other microvolume spectrometer, it took that same person 63 minutes to do all 96 measurements. And if you would have a fibrate system, it even took an hour and a half. And in addition to the capabilities we've discussed so far, Lunatic is also great working in larger scales. For high throughput needs, the plates used for Lunatic are SBS standard and are so compatible with liquid handling robots. The system can be fully integrated with our API setup, so the robot does all the work from you. From loading the sample in the plate, to setting up the experiment, to opening the door, loading the plate in the system, to eventually getting the reports into your LIMS system. And on top of that, you can use plate tracking with barcodes, ensuring traceability. And our features don't stop here. We have IQ procedures available, and for labs doing manufacturing, quality control, or quality assurance, Lunatic has a performance verification tool. These tests with certified cuvettes meet the United States and European Pharmacopoeia guidelines for UV-Vis spectrometers. The tests and all necessary calculations are handled automatically by the software. And finally, Lunatic software is also 21 CFR Part 11 compliant with full audit trail and electronic signature capabilities to help you achieve full integrity on your experiments and reports. So just like how we're getting close to having super fast and easy internet literally everywhere, super fast speed and simplicity has come to DNA quantification. Get the fast, simple, and accurate DNA quant you deserve with Lunatic.